In this video, I want to review a theory for why we age and what we can do about it. So you can take any animal in the animal kingdom, and you can take any one of their cells, and if you look inside of their cell, you'll find this Mtor complex. So what does this Mtor complex do? Well, to simply put it, the Mtor complex senses the energy status of the cell, and the Mtor complex regulates the cell cycle. So essentially, if the Mtor complex senses that the cell's in a high energy state, it's going to stimulate DNA replication and cellular growth and proliferation. However, if the Mtor complex senses that the cell's in a low energy status, it's going to stop the cell cycle, stop cellular growth and division, and it's going to upregulate cellular repair and DNA repair, and also upregulate autophagy. So the fact that every animal in the animal kingdom has this Mtor complex shows that this is a very important system. Also, something important to realize is that in the aging community, this Mtor complex is synonymous with healthy aging. And it's well known and established that excessive Mtor activation accelerates aging. However, if you can blunt Mtor activation, you can extend longevity. So why is this the case? What is happening here? So to understand this, first, let's understand hydrobiology. So this hydra is well known in the aging community for essentially being biologically immortal. This hydra can live indefinitely without aging. So how does it do this? Well, it's related to stem cells. So this is simplifying the story, but the hydra has very powerful stem cells, and these stem cells can constantly renew and produce fresh new stem cells. And not only that, these stem cells can produce specialized cells, specialized somatic cells that each have their own specialized function. However, what's interesting is eventually these specialized cells will be removed and then replaced with a fresh new specialized cell. Then eventually another cell will be removed and replaced with a fresh new cell. And within about 1 month, every single cell within the hydra will be replaced with a brand new fresh cell. So therefore the hydra can do this indefinitely. And it also has very robust DNA repair systems, but as long as the hydra can constantly produce new, fresh somatic cells, there's no problem. And therefore, the hydra essentially does not age, and it can do this indefinitely. So humans are a little more complicating, but generally speaking, for most mitotic tissues, there are stem cells that produce the somatic cells that make up that tissue. Of course, there are examples for post-mitotic cells, but generally speaking, this is the general scheme. So for most tissues, there are stem cells that are maintaining the cells that make up that tissue. And these stem cells can divide indefinitely because they express telomerase. However, it's a little more complicating, so we'll talk about that more in detail later. But generally speaking, the idea is that these stem cells can rejuvenate and produce new, fresh stem cells, and they can also produce specialized somatic cells. And then these somatic cells divide and produce all the specialized cells that make up the tissue. However, what's important to realize is every time one of these somatic cells divide, the telomeres in the chromosomes become shorter. And this is due to the way that linear chromosomes divide, intrinsically due to the mechanism that's used to divide linear chromosomes. The telomeres will get shorter every time the cell divides. So therefore, as these somatic cells divide, every time they divide, the telomeres are going to get shorter. And eventually the telomeres become so short that they activate a signal that triggers the cell to become senescent. This is referred to as replicative senescence. So these senescent cells are extremely important, and they're a very hot topic in aging research. And it's well known that senolytics, which kill senescent cells, extend not only lifespan but also health span. So there's this concept in the aging community where you want to promote health span, where ideally you would like to live to old age and then quickly die. However, normally what happens is people age poorly. So as they age, they spend the last half of their life in bad health. However, when you kill these senescent cells, you extend health span, the amount of time that an organism is healthy, and then as they get very old, they quickly die. And there's also a known concept where these senescent cells are responsible for a lot of age-related diseases. So a lot of people like to think of the diseases in old age as the linear accumulation of damage throughout life. However, we're starting to believe that this is not the case. There is not a linear accumulation. And in fact, what happens is older tissue becomes dysfunctional, perhaps because of these senescent cells. And as older tissue becomes dysfunctional, there's an accelerated formation of pathology. So why are these senescent cells so destructive? Well, it's known that these senescent cells secrete certain cytokines, and this is referred to as the Sasp, the senescence-associated secretory phenotype, where these senescent cells release cytokines, and these cytokines are very destructive. Essentially, they affect the neighboring cells in the tissue. They lead to the stem cells becoming exhausted. They cause more somatic cells to become senescent. So then you have a bunch of non-dividing post-mitotic cells just sitting around accumulating damage. You're not producing new, fresh somatic cells. Rather, you have a bunch of non-dividing post-mitotic cells just sitting around accumulating damage. And also it's known that the Sasp is very strongly pro-inflammatory and also downregulates cellular repair programs. So you can see as these senescent cells start to develop how the tissue becomes dysfunctional, and this leads to physiological deterioration. So we know as somatic cells divide, the telomeres get shorter, and once they reach a certain threshold, that activates replicative senescence. However, telomere attrition is not the only source of cellular senescence. There are other factors that lead to somatic cells becoming senescent. For example, DNA damage is another big factor that can take a healthy somatic cell, and if it has enough DNA damage, that can prematurely form senescent cells. Also, mitochondrial dysfunction and cellular stress. So there are a lot of forms of cellular stress that can take a healthy somatic cell and have it prematurely turn into a senescent cell. And really the way you should think about this is when you're young tissue has a lot of fresh somatic cells and stem cells maintaining those somatic cells. But as you age, you start to accumulate these damaging senescent cells. Fortunately, our immune system can come along and kill those senescent cells. However, as you age, because of lots of different factors, you're going to accumulate more senescent cells. And as you age more, you're going to accumulate more senescent cells, and eventually you're going to overwhelm the immune system. And it's known that when you're very old, most of your cells have become senescent. So this is a signature of old dysfunctional tissue. You have a lot of senescent cells and you have a lot of immune cells, and these immune cells come for various reasons. And keep in mind, these cells are not dividing. They're post-mitotic senescent cells, so they're not dividing, so they're just sitting around accumulating damage. And this leads to dysfunctional tissues and physiological deterioration. So why should we care about cellular senescence? Well, remember, these senescent cells secrete a senescence-associated secretory phenotype, where they secrete these very destructive cytokines. But it's important to realize that the cytokines that are secreted depend on the type of tissue and why that tissue became senescent, and also the time course. For example, is it early on once the cell becomes senescent, or is it later on? But the general theme is that this Sasp, this senescence-associated secretory phenotype, is very destructive to neighboring tissues. So there's a lot of research done on how to kill these senescent cells. So what exactly does a Sasp do? Well, it's known to affect stem cells, leading to stem cell exhaustion. And remember, it's these stem cells that are producing fresh new somatic cells. So if you cause stem cell exhaustion, these somatic cells are going to become senescent, and you're just going to have a bunch of non-dividing cells accumulating damage. But it's important to realize that there are many complex factors that lead to stem cells becoming exhausted. For example, there are interesting experiments referred to as Parabiosis Experiments, where you take a young person and connect their blood to an old person. And it's known when you do these experiments that soluble factors in the old person enter into the young person and cause the young person to have increased senescent cells, and also causes the young person's stem cells to become exhausted. However, the reverse is shown, where soluble factors in the young person can enter the older person and improve stem cell function. And in fact, we think we know what those factors are, for example, GDF11, and which is actually used for a lot of therapies. So the idea is it's these Sasp cytokines which leave the senescent cells of the old person and enter into the young person and induce cellular senescence and stem cell exhaustion. And this is a big deal because, again, it's these stem cells which produce fresh new somatic cells which can divide, producing fresh cells. However, if you inhibit stem cell function, then you're going to stop producing somatic cells, and these somatic cells are going to become senescent, and you're just going to have a bunch of non-dividing post-mitotic senescent cells just sitting around accumulating damage. However, this Sasp has other destructive effects on neighboring somatic cells. For example, there are these really important sirtuin enzymes, which are found in virtually all cells in the animal kingdom. And these sirtuin enzymes are very important for DNA repair. In fact, we can see that they're involved in virtually every single form of DNA repair there is. But not just that, these sirtuins also are very important for regulating gene expression. They essentially regulate the epigenome, and they're also very important in activating very important transcription factors, which are important for DNA repair, reactive oxygen species removal, autophagy, mitochondrial biogenesis, and the expression of antioxidant genes. And they're also known to inhibit NF-kappa beta, which is a very potent pro-inflammatory transcription factor. So we can see how important these sirtuins are. And in fact, these sirtuins are synonymous with upregulating cellular repair programs. And they're also known to catalyze over 500 different chemical reactions. But what's important to realize is for these sirtuins to function, they need the NAD molecule. They need the NAD molecule and essentially break down the NAD molecule every time they catalyze the chemical reaction. And also what's important is oleic acid in olive oil and resveratrol in red wine is known to activate the sirtuins. But even if you activate the sirtuins, the fuel that the sirtuins need to do their chemical reactions is NAD. So they need this NAD molecule to do their chemical reactions that lead to cellular repair programs. And what's known is that this Sasp activates CD38, which destroys NAD and lowers NAD. And we know that as an organism ages, the NAD in their cells decrease. So therefore, these sirtuins can't function and they can't do all their cellular repair programs. So this is a very curious phenomena where these Sasps essentially intentionally downregulate cellular repair programs. And just keep in mind that NAD is extremely important. NAD is also used by a completely different set of enzymes that catalyze DNA repair. And it's also known that as NAD decreases, that leads to mitochondrial dysfunction and many downstream consequences. So these senescent cells are essentially intentionally downregulating cellular repair programs, which leads to somatic cell dysfunction. But not only that, it's known that this Sasp phenotype secretes extremely potent pro-inflammatory cytokines. And a known signature of old dysfunctional tissue is a decreased lymphocyte to monocyte ratio, where essentially you have fewer adaptive lymphocytes, but you have an increased amount of innate immune cell monocytes. And this decreased ratio is associated with many age-related diseases like cancer, cardiovascular disease, pulmonary disease, and all-cause mortality. And it's known studying the genetics of centenarians, people who live to 100 and beyond, they have less inflammation. And this has led to the term of inflammaging. So the point is that we know that the immune system is extremely important when it comes to aging. Also, what's interesting is a lot of aging researchers believe that the thymus may play a causal role in aging. For example, it's known that the thymus is responsible for producing new, fresh, naive T cells, which are very important for adaptive immunity. However, as we age, the thymus degenerates, so therefore we have a weaker, decreased adaptive immunity. However, experiments that have shown that once you restore the thymus tissue, that restores this ratio to youthful levels. And not just that, a lot of interesting things happen when you restore thymus tissue. For example, PD-1 decreases. And to put that into perspective, pharmaceutical companies spend around $14 billion per year on PD-1 because of its role in cancer. And not just that, PSA decreases, prostate-specific antigen. And it's known that the prostate becomes dysfunctional and prostate cancer increases as we age. So therefore, prostate health is a good proxy for aging. So the idea is that when we're young, we have a robust response against specific antigens and a very strong adaptive immune system. However, as we age, this adaptive immune system fails, and that's why a lot of older people die of infections. And not just that, essentially, as we age and as the adaptive immune system decreases, that leads to broad inflammation and uncontrolled innate immunity and inflammation. And it's also known that these Sasp cytokines go to neighboring cells and convert those neighboring somatic cells into senescent cells, forming new senescent cells. And also, this Sasp is associated with increased cancer formation, although that mechanism is very complicating. So the general idea is that for most tissues, we have stem cells, which produce somatic cells. And these somatic cells can divide and produce new, fresh somatic cells. So even if you damage one of these somatic cells, it's okay because they're going to divide and produce new, fresh somatic cells. But as these somatic cells divide, eventually we're going to form senescent cells. And we know these senescent cells inhibit stem cells, and they trigger somatic cells to form new senescent cells. So then you begin to accumulate non-dividing post-mitotic senescent cells, which just sit around and accumulate damage. And not just that, these senescent cells down-regulate repair programs. So then things just go downhill from there, and that leads to physiological deterioration. So the way I like to think about it is we have a rejuvenation phase where we have mitotic cells producing new, fresh cells, which eventually leads to this deterioration phase, where you have essentially non-dividing post-mitotic cells just sitting around and accumulating damage. And perhaps it's our genome that regulates the rate that we progress through these phases. For example, maybe some organisms have very robust stem cells. Or they have somatic cells with very long telomeres, so therefore they can divide for a long time before forming senescent cells. Or they have very good cellular repair programs like DNA repair and reactive oxygen species, which postpones the onset of these senescent cells. Because remember, there are many different ways to form senescent cells. Or perhaps their genome regulates their immune system in different ways. The idea is that perhaps it's our genetics that is the primary factor controlling longevity and controlling the progression and the rate through these phases. So therefore, how would we extend longevity? Well, again, we're stuck with our genetics. So we want to maximize our genetics and decrease the limitations that our genetics give us. So therefore, the idea is we would want to slow down progression through these phases. And we would want to attenuate progression through this rejuvenation phase. Because while we're in this rejuvenation phase, our tissues are very resilient. Even if we damage tissue, it's okay. Our stem cells are going to produce new, fresh somatic cells. So our tissue is very resilient. And we would also want to postpone the onset of this deterioration phase, essentially postpone the onset of senescent cells and reduce the damage caused by these senescent cells. So how would we do this? So this brings us to mTOR. So what does mTOR have to do with longevity? Well, something interesting is there's a prize given to the researchers who can make the longest-lived mouse. So what was the longest-lived mouse? Well, it was a mouse that was genetically modified to have a growth hormone receptor knockout. So why? Why did knocking out growth hormone receptor lead to the longest-lived mouse? Well, it's related to mTOR. So mTOR is a key regulator of the cell cycle and cell division. And essentially the idea is if you activate mTOR, you increase the rate of the cell cycle. So therefore, theoretically, you would increase the rate you go through this rejuvenation phase. And what are the things that regulate mTOR? Well, you need two things to activate mTOR. First of all, you need amino acids, primarily leucine, but there are other amino acids. But you also need growth hormone, which leads to IGF. And also insulin, which leads to increased activity of IGF. So the idea is if you're constantly activating mTOR, you're increasing the rate of the cell cycle and cell division, so therefore you would increase the rate you go through this rejuvenation phase. So the idea is the more you activate mTOR, you age faster. You essentially age in times 2 speed. However, if you inhibit mTOR, you age slower, you age in 1/2 speed. And that's essentially what happened with this growth hormone receptor knockout. These mice had very low activity of mTOR, so therefore there was a low rate of the cell cycle, so therefore theoretically they went through this rejuvenation phase in slow motion. But it's a little more complicating than that. So before we mentioned how stem cells can divide indefinitely because they express telomerase. But the truth is, yes, certain like embryonic stem cells express telomerase, but most stem cells in most tissues only have very low expression of telomerase. So even as these stem cells divide, their telomeres do shorten because, yeah, they express telomerase, but they only express low levels of telomerase. So as these stem cells divide, their telomeres are shortening, but they're shortening at a lower rate. But eventually, these stem cells will become exhausted and will stop dividing. So the idea is that there's a battle between telomerase and mTOR in these stem cells. Essentially, telomerase is increasing the length of the telomeres. However, mTOR is telling the stem cell to divide, and every time it divides, the telomeres get shorter. So there's this battle. So the level of telomerase versus the activity of mTOR essentially determines the rate of telomere shortening, which will increase the rate of stem cell exhaustion. So this may be another reason why inhibiting mTOR led to longevity. And there are a lot of molecules that are known to activate telomerase and to increase telomeres, but it's a little too early to know if these will lead to increased longevity. Also, what's interesting is it's known that metformin and rapamycin inhibit mTOR, therefore decreasing the rate of cell cycle and cell division, theoretically decreasing the rate that we go through this rejuvenation phase. But not just that, metformin and rapamycin inhibits mTOR, which through complicated mechanisms also lead to increased autophagy. So what is autophagy? Well, we know cells have organelles. However, these organelles become damaged. We have worn out organelles, for example, mitochondrial dysfunction, worn out ribosomes, and ER. And these worn out cells accumulate damage. And also the molecules in these organelles cross-link, and you have the buildup of misfolded proteins, so you have dysfunction on a cell level. However, when you activate autophagy, you essentially take these worn out organelles and you essentially destroy them and use them as a source of energy to produce new, fresh organelles. And that leads to better cell function. So the idea is that metformin and rapamycin inhibit mTOR, which leads to increased autophagy. So essentially, it's like a spring cleaning for a cell, taking all the worn out, damaged organelles and destroying them and producing new, fresh organelles. Also, there's really interesting data that shows that metformin lowers the cancer incidence on a wide range of cancers, which is very interesting. So why? Why does metformin lower the cancer incidence on many different types of cancer? Well, remember, when you activate mTOR, that increases cell division. So if you're doing a lot of cell division, there's an increased chance of producing chance mutations, which can theoretically lead to cancer. So that's one theory for why metformin lowers cancer incidence. But I actually have my own theory, and again, it's just a theory. This hasn't been proven. But the idea is that every cell is debating whether it wants to do anti-apoptosis or pro-apoptosis. It's debating whether it wants to commit apoptosis. But something important to realize is when a cell has damage, it should induce apoptosis. However, when you're constantly having growth hormone and IGF and activating mTOR, you're promoting growth. You're promoting cell growth and cell division, and therefore you're giving that cell anti-apoptotic signals. And this is actually bad, because if a cell has damage, if it has DNA mutations, and it has other forms of cellular stress and cellular damage, then it should commit apoptosis. However, if it's constantly getting a balance of anti-apoptotic signals, then it will not commit apoptosis and it will keep dividing, and that's not good. However, when if a cell has damage, then it should commit apoptosis. It should have strong pro-apoptosis signals so it doesn't divide to produce new dysfunctional cells. And that's one theory why metformin may decrease the cancer incidence. Essentially, metformin balances out giving pro-apoptotic signals. So therefore, if the cell has damage, however, if you're constantly giving it pro-growth signals, then it's going to continue to produce new cells. However, if the cell has damage and you're taking metformin, it's going to give it pro-apoptotic signals, so then the cell will die and it won't produce new dysfunctional cells. Also, there's a syndrome referred to as Laron syndrome. There's a population in South America where essentially they have an IGF mutation. So essentially their IGF isn't working, and they're known to have dramatically lower cancer incidence. So essentially it's the opposite of having high IGF. If someone has high IGF, that's pro-growth signals. That's pro-growth even if you should not grow. But Laron syndrome is essentially the opposite. It's low IGF. So therefore, if you have Laron syndrome and one of your cells has damage, then it's going to have the right pro-apoptotic signals and it's going to destroy itself. Also, vitamin D is also known to be pro-apoptotic and may be responsible for why vitamin D leads to lower cancer incidence. And also EGCG, one of my favorite molecules, it stabilizes P53, essentially creating a robust pro-apoptotic signaling. And green tea, which has EGCG, has been known to lower cancer incidence. And perhaps that's why, because when you drink green tea, you have EGCG stabilizing your P53, creating a very robust pro-apoptotic signaling pathway. So therefore, if a cell is bad and has damage, then it will be pro-apoptotic. Also, this is a little random, but we know elephants have many cells. So therefore, there should be a high chance that just one cell becomes cancerous and the elephant dies. However, it's known that elephants have lower rates of cancer. And one theory is because elephants have 20 copies of P53, and therefore they have a very robust pro-apoptotic signaling pathway. So again, this is just a theory that if you want to lower cancer incidence to promote a robust pro-apoptotic signaling pathway, and that's a theory for why metformin works, because essentially metformin tells the cell that it's in a low energy state and activates AMP kinase, which then leads to pro-apoptotic signaling pathways. So therefore, if a cell has mutations and damage, forms of cellular damage, it's primed to be pro-apoptotic and to die, so it doesn't produce damaging cells which can lead to cancer.